Welcome to the deep dive. Today we're sliding into the molecular world of fats, or maybe more precisely, as the biochemists call them, lipids. We've dug into that pile of biochemistry sources you sent over the learner and really tried to distill them down. Our mission today, let's unpack the basic structures and, honestly, the pretty amazing versatility of lipids. They're kind of the unsung heroes, right? Handling energy, building cells, even running signaling systems.
That's a great way to put it. And fundamentally, when we talk lipids, the key thing, the starting point, is they are hydrophobic. They just don't mix well with water. So insoluble in water, but they dissolve easily in, organic solvents. And because of that property, they end up in three main places in your body: floating around in plasma, stored in adipose tissue, or forming the structure of biological membranes.
Okay, so if they all share that water-hating trait, how do the sources categorize them? Seems like they fall into two main camps. You've got the simple lipids, mostly about energy and protection, things like fatty acids, triacylglycerols, waxes, and then the complex lipids, which sound like the more specialized ones, the architects and messengers. That's where phospholipids, glycolipids, and steroids come in.
Exactly. That's a good functional breakdown.
All right, let's unpack this then. Starting right at the foundation, fatty acids. The simplest units, but wow, they seem to dictate so much about the bigger picture.
You really do. Structurally, you're looking at a carboxylic acid, that's the head, with a long hydrocarbon chain attached, the tail. And what's really neat biologically is the specificity. These chains are almost always straight, unbranched, and have an even number of carbons, usually 16 or 18.
An even number, why is that?
It comes down to how they're built in the body. They're synthesized using two carbon building blocks, acetate units, essentially. So you naturally end up with even numbers.
Makes sense. And the key feature seems to be saturation. That tells you how they behave physically.
Precisely. Let's start with the straight ones, saturated fatty acids. No double bonds in the chain, just single bonds all the way down. Think of stearic acid, 18 carbons, totally saturated. Because it's so linear, these molecules can pack together incredibly tightly.
Like stacking pencils, perfect.
Yeah,
right?
exactly. That tight packing means strong forces between molecules, which translates to a high melting point. Stearic acid melts way up at 70 degrees Celsius. And that's why your animal fats, rich in these, are solid at room temp.
Right. Okay, but here's where it gets really interesting, I think. Introduce just one double bond, and you get the unsaturated fatty acids. The geometry changes everything.
It totally does. And crucially, in nature, these double bonds are almost always in the cis configuration. This isn't just a minor tweak. It introduces a significant kink, a bend, in that straight chain.
And that kink is the whole story, isn't it? Trying to stack those kink chains is like trying to stack, bent sticks. You just can't pack them tightly.
Exactly right. There's space between them. And that inability to pack closely, that disorder, drastically lowers the melting point. Take oleic acid, same 18 carbons as stearic, but with just one cis double bond. The melting point plummets down to 13 degrees Celsius.
Wow.
That's why your plant oils, full of unsaturated fatty acids, are liquid at room temperature. It's pure structural chemistry.
And this structural difference connects right back to health, doesn't it? We should probably touch on trans fatty acids here.
Oh, definitely. We have to. Because while the natural cis bond creates that kink, trans fatty acids are different. They often pop up during industrial processing, like partial hydrogenation that's turning liquid oil semi-solid for things like margarine.
So what's the deal with the trans bond?
Well, the trans double bond doesn't introduce that sharp kink. The molecule stays relatively straight, more like a saturated fat.
Ah, I see. So even though it's technically unsaturated, its shape lets it pack tightly.
Exactly. It mimics the structure and therefore the packing properties of a saturated fat. And that's why they're linked to health risks like higher LDL cholesterol and atherosclerosis. The body seems to handle these somewhat unnatural straight shapes less effectively.
That's fascinating. The body's reading the shape, not just the GON count. Okay, so we've got the building blocks. Where does the cell store this stuff for energy later?
Right. That brings us neatly to the main storage form, triacylglycerols. You probably know them as tags or triglycerides. Very simple concept. It's just three fatty acid molecules esterified or chemically linked to one glycerol molecule backbone.
And these are packed away in our adipose tissue, our fat cells.
Correct. That's their primary residence. And their main job, super efficient energy storage.
How efficient are we talking, like compared to carbs?
Oh, way more efficient. Think about energy density. Carbs and proteins give you about 4 kilocalories per gram, dry weight. Lipids, partly because they're more reduced chemically and don't carry associated water, pack a huge punch, about 9.3 kilocalories per gram.
Yeah, more than double.
Yeah. That density is why an average person can store enough energy in their fat reserves to last for weeks, potentially. Your glycogen stores, the carb reserves, you'd burn through those in less than a day.
That density really makes them vital. There was a quick chemical note in the sources, too. Saponification. Sounds like soap making.
It literally is. If you take tags and react them with a strong base, say sodium hydroxide, you break those ester bonds. You get back the glycerol and you get the sodium salts of the fatty acids, and that mixture is soap. It's a neat link to their basic chemistry.
Huh. Cool.
Yeah.
And connecting this back to everyday life, the sources brought up the classic butter versus margarine debate.
Right. Butter naturally is loaded with those tightly packing saturated fatty acids and also cholesterol. Margarine typically starts from plant oil, so it's richer in the kinked, unsaturated fatty acids.
But it's not that simple health-wise, is it? Because of the processing.
Exactly. The traditional way to make margarine solid involved that partial hydrogenation process we mentioned. And, that generated those unnatural trans fatty acids. So you started with potentially healthier unsaturated fats, but the processing introduced a different kind of risk, one that structurally behaved like saturated fat anyway.
So the processing really muddied the waters on which was better. It shows how much structure matters.
Totally. Structure dictates function and interaction in the body.
Okay, so tags are the fuel depot.
Yeah.
Let's switch gears to the lipids that actually build things, the architects. How do we get from simple fuel to cell membranes and phospholipids?
This is where lipids get really elegant, I think. It comes down to the amphipathic nature of phospholipids. It's a bit of a mouthful, but it just means they have two different personalities towards water.
Like a molecular Jekyll and Hyde?
Sort of, yeah. They have a polar head group, usually containing phosphate, which is charged and loves water. It's hydrophilic. And then they have typically two nonpolar fatty acid tails, long hydrocarbon chains that hate water. They're hydrophobic.
And that dual nature is perfect for making boundaries.
It's perfect. When you toss these molecules into water, they spontaneously arrange themselves. The hydrophobic tails all turn inward, hiding from the water, clustering together. The hydrophilic heads face outward, interacting with the water inside and outside the cell.
Forming a sheet like a sandwich?
Exactly. A double layer, the lipid bilayer. That is the fundamental structure of every biological membrane. It forms automatically because of those opposing properties.
And we know these membranes aren't rigid walls, right? They're dynamic. The fluid mosaic model comes to mind here.
Yes, Singer and Nicholson, 1972. That model is absolutely key. It depicts the membrane as this fluid sea of lipids, with various proteins either embedded within it, integral, or associated with the surface, peripheral. It's not static.
So things moving around.
Constantly. The lipids themselves and many of the proteins are free to move laterally, side to side, within their own layer of the bilayer. This fluidity is essential for membrane function, letting things interact, allowing the cell to change shape, and so on.
And what controls that fluidity? The sources highlighted cholesterol. We hear so much negative stuff about cholesterol, but in the membrane, its role seems purely positive.
Within the membrane, its role is absolutely essential and, yes, beneficial. Think of it as a fluidity buffer, kind of like antifreeze and thickener combined.
How does that work?
Well, at normal or higher body temperatures, the rigid steroid ring structure of cholesterol wedges between the phospholipid tails and actually restricts their movement a bit, preventing the membrane from becoming too fluid, too leaky.
Okay.
But then, at lower temperatures, cholesterol does the opposite. It disrupts the tight packing of the fatty acid tails, preventing them from getting too close and solidifying or crystallizing. It keeps the membrane fluid even when it gets cold.
So it maintains that just right Goldilocks data viscosity? That's clever.
Very clever. It's vital for membrane integrity across different conditions.
And besides the main phospholipids, there are other specialized structural lipids too, right? Like sphingolipids?
Yes. Sphingolipids are important. They're built on a different backbone molecule called sphingosine, not glycerol. A really critical example is sphingomyelin. This is a major component of the myelin sheath.
The insulation around nerve cells.
Exactly. That sheath is crucial for rapid nerve impulse transmission. And get this, the myelin sheath can be up to 80% lipid by dry weight. Sphingomyelin is a huge part of that insulating layer. It really highlights how specialized lipid structures create specialized functions.
80%. Wow. Okay, let's make one final shift. We've done energy storage, we've done structure. What about the lipids that act as signals regulating the body.
Right, the messengers. We can start with the steroids. These are structurally very different. They aren't built from fatty acids in the same way,
Yeah.
and they can't be easily broken down by simple hydrolysis. Their defining feature is that core structure. 4 fused carbon rings, making them very rigid and flat. It's called the gonane nucleus.
Why is being rigid and flat useful for a hormone?
Well, think about what hormones need to do. They often travel through the bloodstream, then need to pass through the target cell's membrane, which is itself lipid-based, to reach receptors often located inside the cell. That flat, somewhat greasy, rigid structure is pretty good at slipping through membranes and fitting into specific receptor sites.
Ah, like a key fitting into a lock that's embedded in the wall.
Kind of, yeah. And the parent molecule here, the precursor for all other steroids, is cholesterol again. Shows how central it is. From cholesterol, your body makes all the essential steroid hormones like cortisol for stress response, testosterone, estrogen, plus the bile acids needed to digest fats, and even vitamin D, synthesized in the skin with sunlight.
So cholesterol is the starting point for a whole class of regulators. What about other signalers, the eicosanoids?
Eicosanoids are fascinating. They act locally. They're often called paracrine hormones. Unlike something like estrogen that travels systemically, these are made, act on nearby cells, and are then broken down very quickly. They're all derived from a 20-carbon polyunsaturated fatty acid called arachidonic acid.
So they're for immediate local effects.
Exactly. Think inflammation response, blood pressure regulation locally, smooth muscle contraction, blood clotting, pain signals. They mediate those kinds of rapid neighborhood responses. Examples include prostaglandins, thromboxanes, and leukotrienes.
If they're involved in inflammation and pain, is that how drugs like aspirin work?
Precisely. That's a perfect example. Aspirin works by inhibiting COX enzymes, the enzymes that synthesize prostaglandins from arachidonic acid, block the synthesis of the signal molecule, and you reduce the downstream effects like pain and swelling. It shows how potent these local lipid mediators are.
Very cool. Okay, and finally, the sources mentioned the fat-soluble vitamins are also lipid-derived.
Correct. Vitamins A, D, E, and K are all lipids, technically isoprenoids. They're built from repeating 5 carbon units, similar precursors to cholesterol in some ways, and they have incredibly diverse regulatory roles, too. Vitamin A in vision, vitamin E as a key antioxidant protecting membranes from damage, vitamin K essential for the blood clotting cascade. It just underscores the incredible functional range lipids cover.
This really has been a deep dive. It's amazing how that one simple property-hating water leads to such diverse roles. So, quick recap. Lipids give us dense, long-term energy storage with triacylglycerols. They build the fundamental structure of our cells with phospholipids and cholesterol acting as modulators. And they serve as critical signals, both system-wide, like steroids, and locally, like eicosanoids.
Absolutely. And, tying this back to the structural lipids, especially those complex ones like the sphingolipids we mentioned in myelin, it raises a really important point about balance. Building these things is only half the story.
What's the other half?
Breaking them down. Our cells rely on constantly turning over and recycling these complex molecules. The membranes aren't static forever. Components get old or damaged and need to be replaced.
Oh, okay.
And there are some really devastating, though rare, genetic diseases, things like Tay-Sachs disease or Niemann-Pick disease. These are caused by defects in specific enzymes, usually in the lysosome, the cell's recycling center. These enzymes are supposed to break down specific sphingolipids.
But if the enzyme is broken,
the breakdown fails. The lipid just accumulates, builds up inside the lysosome, eventually crowding the cell and causing massive dysfunction, particularly severe neurological damage, because the brain is so rich in these lipids.
wow. So it's not just about making the right structures, but also having the cleanup crew working perfectly. That really highlights the cell's constant maintenance work. What stands out to you thinking about this, about that delicate balance required, not just building complex lipids, but needing the systems to constantly and efficiently dismantle them too, just for basic cellular health. Something to chew on next time you just hear the simple word fat.